Well, hello, good morning, good afternoon, good evening, everybody, depending on where you're joining us from the rest of the world. My name's Derek Jones, and it's my real privilege and pleasure to be able to host this, the Diffusion Campfire, historical campfire. Now, this is a celebration of the evolution of our field of diffusion MRI, and it's my pleasure to be in such diffusion royalty that I'll introduce in a second. I'm going to talk about a critical time in the field of diffusion MRI. And there was a prophecy from 72 years ago from Erwin Hahn in this classic paper on spin echoes. And if you look in the abstract here, you can see that Professor Hahn talked about the effect of diffusion on relaxation measurements. So he prophesied that something was going to happen, and that was in 1950. Now, George Orwell died in 1950, but just a year before he did, he wrote another book, 1984, and this prophesied a dystopian world where the world was miserable to live in and things were not very pleasant indeed. How accurate was he? Well, let's have a quick look. What happened in 1984? Well, in January, we saw Apple Macintosh computer go on sale, priced at $2,500. The This Is Spinal Tap mockumentary was released in the US, and just a bit of trivia, Derek Smalls, that appears in the video, has since recorded a song called MRI, so you can Google that and watch him sing about that. Virgin Atlantic launched its first flight from London to New York. The Summer Olympic Games were opened in Los Angeles. A Nightmare on Elm Street was released in the United States, which was pretty horrific. But also, perhaps more notedly, Princess Diana gave birth to Henry Charles Albert David, or Harry. So whether Orwell was right or wrong about 1984 depends on your point of view. But there were some other really good things that happened in 1984. So in a corner of San Francisco, there was a group of 3 intrepid scientists, George Wesbey, Mike Moseley, and Dick Ehman, that were looking at the effects of diffusion on relaxation measurements and observed that there was a reduction in biological T2 that they attributed to diffusion. Now, they were making these measurements on an imager and were able to make the first in vitro images of the self-diffusion coefficients. And these are some of the very first in vitro images. In another corner, this time in the UK, we have Mary Claire Bushell doing a PhD with David Taylor. And in chapter 7 of her thesis, you can see here, she's already started to think about measuring diffusion MRI. And this is the post-gradient spin echo experiment in which he uses this to make an image of the ADC in the egg. And this paper was subsequently published with Taylor and Bushell. Then we see this sequence again from another corner, this time in Orsay in France, with Denis Bihan and Eric Breton in this paper written in French in Comp Rendu. But luckily for us, there's a translation. The authors present an original NMR imaging method to obtain a calculated spatial distribution of molecular diffusion coefficients. And here again, we see the sequence. And here, a treatise on the contribution of the various imaging gradients, allowing us to get accurate measurements in the diffusion experiments. We go back to San Francisco in 1990, where Mike Moseley had already looked at the drop in diffusion in acute ischemia in cats, but observed something new and exciting, which was the anisotropic diffusion in the cat central nervous system. First, just showing it happens in sugarcane, but then looking at these beautiful delineations of anisotropy of white matter. And one of my favorite images, the clear orientational specificity in the spinal cord. But to make these images more practical, a quantum leap was developed by Bob Turner, who introduced echoplanar imaging into diffusion MRI. And with the sequence that we now all know and love and is ubiquitous in labs around the world. Well, this was at the NIH, where we now return to the anisotropic diffusion, and Peter Basser enters the forum, working with Denis, looking at fiber orientation mapping in an anisotropic medium. And these were abstracts presented at the ISMRM in Berlin in 1992, and estimating the full diffusion tensor, which then led to these two classic papers, the estimation of the tensor and actually imaging the tensor, allowing us to see these in vivo images of the diffusion ellipsoids. So you can see it was, in fact, a fantastic decade from 1984 to 1994. And I've given you my brief perspective, but it's far better to hear from the experts. And so it's my pleasure to introduce our speakers today. We have kicking us off will be Mike Moseley and Dick Ehman. So if you'd just like to introduce yourselves.
Yeah, it's Mike Moseley, and you'll see across the screen Richard and Dick Ehman. We were at UCSF during the exciting time of the birth of the Diasonics scanners, and there was a little, I think it was a 100 megahertz scanner that Larry Crooks and Leon Kaufman had built, and that we were using back in 83, 84, when all of this got started. Dick?
So yeah, so I was a research fellow at UCSF in 84, 83, 84, and had this wonderful opportunity. Timing is everything. This wonderful opportunity to be involved in the early exploration of MRI at that location.
Thank you. And then on to Denis.
Yes, long ago, more than 30 years ago. Well, I was at the time a medical resident in neurosurgery and neuroradiology, and at the same time, PhD students in nuclear physics and elementary particle physics, so totally different things. But I was very lucky that I got invited to work on the very first MRI scanner built in France by the company called Thomson CGR at the time. So I was very lucky to have 0.35 Tesla system. And this is how I got my fingers hooked to develop diffusion MRI as you have presented.
Thank you, Denis. And then we know you were joined at some point by Bob. So over to Bob Turner.
But yes, I'm Bob Turner. At the time in 1984, I was a temporary lecturer at the University of Nottingham. I just basically joined Peter Mansfield, already with a desire to try to use MRI to study human brain function, but no idea how to do it. And also, I was just learning MRI. I started to learn MRI in 1984 basically at Peter Mansfield's feet. So it was, but I had a scanner of my own to play with, which was 0.1 Tesla, on which I was able to actually write my own EPI sequence. It's a home-built system, really. Well, that was that. Yeah. The rest of the story will come along later.
Sure, thanks, Bob. And last but definitely not least, Peter Basser.
Thank you, Derek. Well, in 1984, I was, still in high school. No, I'm just kidding. But I was actually, doing my doctoral thesis in 1984. I'm a little late to the game because I got involved in diffusion imaging, in 1991. But once I got to the NIH after getting my PhD, I was working on a few problems with involving transport of mass and transport of charge, convection enhanced delivery and magnetic stimulation. And both of those problems required heterogeneous anisotropic maps of material properties. So I was kind of primed to find a methodology like diffusion imaging that would provide that type of information. So we'll probably talk more about things later on, but I think that's a good start.
Great, thanks, Peter. And notably, a couple of you talked about lower field than we have now. It's interesting how the trend is going now to low field and ultra low field. And Denis talked about a 0.35 Tesla system. And I know that, Mike, that's a system that's close to your heart. So I know that you've prepared some slides and something to tell us a little bit about life in 1984. Maybe you'd want to tell us what you were doing just the year before. I'm thinking if we have trainees that are listening that may be on the cusp of their big discovery in the following year, You know, what were you doing just before this golden era of diffusion?
Yeah, well, I had just finished my PhD in Uppsala, and it was about measuring translational self-diffusion of small molecules inside of biopolymers and stuff using MRI. And of course, Stayskull Tanner was near and dear to my heart. And so I get to UCSF. I decided to take a postdoc with Tom James at UCSF. And I was kind of just wandering around the lab, started in 82, 83, wandering around the lab, wondering what to do. And I was working at the time on a sodium scanner that was being financed by Diasonics in South San Francisco. Diasonics was the manufacturer that kind of messed around with UCSF and was building a whole body scanner. So there I was, you know, trying to wire up the hardware for a sort of a home-built sodium 5.6 Tesla scanner. And this guy, this research fellow from radiology, who was down the hall and a few floors down from me in pharmaceutical chemistry, he kept bugging me. He would come up and he'd go, "Look, I'm George Wesbey. I got a problem. I need to talk to you. I understand you did diffusion for your PhD. Can I ask you a few questions?" And bug me, always popping around. It was like a detective, right? Always there. And his thing was, "Look, I'm trying to measure T2 down in South San Francisco on this Diasonic scanner. The problem is I'm not getting correct T2 values. And I got to ask you, what's going on?" And he would start talking about Edwin Hahn. Now, you know, Hahn was living in the Bay Area at the time. And I don't know if George did meet him, but I don't think it was in 83, 84. But he kept saying, "Look, there's something wrong with that scanner. Can you just take a look?" And I'll show you the, let's see if I can find that. There we go.
What you're doing, Mike, just to say, we tried our very best to contact George. He's working in private radiology at the Scripps Institute. Mike was extremely kind in also reaching out, but I guess he's a busy guy. He's not anymore.
I thought I was going to have to fly down to San Diego to find this guy because, you know, a radiologist in clinics in the Scripps complex, which is like 500 hospitals in San Diego, you couldn't get a hold of him. And you couldn't even leave a message. And I've been trying all week to try to get a hold of George because his infectious enthusiasm was just enormous. When he would show me these pictures, okay, look, I've got these T2 relaxation times and they're not coming out right. And whenever I change the strength of you can see it there, the slice selective gradient, the T2 is changing. What's going on? Well, yeah, at the time we kind of knew, or I kind of knew that this was probably a spin echo sequence. You're using relatively long TEs. I'm going to bet that your hero, Edwin Hahn, would have something to say about this. And that was that it was diffusion. So what we did, well, we made a bunch of tubes and we put them in the scanner. And then we measured the echo amplitude of these various tubes, right? And then we would use water as sort of a reference. And then what we would get would be like a typical plot that you'd see on the right. And we would say, okay, well, when you measure the slope of that plot, you actually were getting the self-diffusion coefficient. And these were well known at the time. We were doing acetone, and we were doing doped water and glycerin and things like that. So we kind of knew what we would be looking for. And that led to the whole idea that in these two papers, let me go back and find them. Yeah, these two papers were submitted to investigative radiology. Now, Dick can tell you at the time, investigative radiology was like, if you didn't publish there, you were just not published. And nowadays, of course, investigative radiology is hard to find. I don't even know if I know it's still around because I've seen a few recent copies, but wow, investigative radiology, I don't think they got a lot of circulation outside of UCSF. These papers did win the AUR Memorial Award, which is the Association of University Radiologists. And it was a big deal. And Larry Crooks down at South San Francisco, I believe, had written a statistical Tanner sequence for the big 0.35 Tesla whole body scanner that he was working on, but he never published anything. I might have shown an image at the SMRM, sorry, but didn't get anything out. And I think it was because of the eddy currents. So that's how it started for us, these two papers. And under the guidance of Dick Ehman, George and I were just like going nuts. Dick, do you remember it that way?
Very good description. My little perspective, Mike, you're very kind. And George had been working with Bob Brash, who was developing looking into contrast agents for MRI. And in fact, George was on the very first paper, along with Bob, of course, and Hans Weinmann, on the use of gadolinium DTPA. And I think the key thing was that we had, as I say, I joined that group in '83 as a fellow, and we had a thing called a Bruker mini spec. It was a relaxometer, and we could measure the relaxivities of aqueous solutions of contrast agents, and we could measure T2 accurately. And then when we tried to do the same thing in the scanner, as you described, the numbers were coming out way too small. Like the longest, the T1 of water was 177 milliseconds, way too short. But anyway, that's kind of, and really George is the one that sought you out and really found this. And then I think that I love the fact that this particular paper here, you really pointed to the direction, as you can see on the screen here, with the idea that this could look at anisotropic diffusion. I'll just stop there, Mike. You might want to expand on that.
Well, anisotropic diffusion was something that I'd known a good bit about because I was doing some of the statistical Tanner stuff in liquid crystals. And of course, at certain temperatures, they would become extremely anisotropic. And it was a great way to study those phase changes. I'm going to stop there and just say that it was a wild time. Everything was happening. And we were taking bus rides down to Diasonics every day to see the latest MRI images coming off of the 0.35 Tesla scanners. But George Wesbey was quite dynamic. I mean, he would not take no for an answer. And he was, he knew what, he was a man on a mission. He knew what he wanted to show. And Edwin Hahn obviously had made a huge impact on him because he was convinced that this is what it was. And indeed, guess what? It's exactly what it was. And those were really exciting times. Well,
it's fascinating, as always, to hear you speak about this. In fact, I was just looking at this is your second paper. And I just like the speculation at the end. And I was going to ask you about this. As I think we may hear later from Denis, you were measuring kind of relative, you were referencing the diffusion coefficient to that of water. But nevertheless, there's a comment here, and again, it's a kind of prophetic. You say, we hope that this measurement will prove to be a valuable and more understandable biophysical parameter measurable by MRI for the non-invasive characterization of biological systems in vivo. I mean, this was really the birth of diffusion imaging. I just wondered, do either of you, what's been the biggest surprise from those initial measurements from what's been achieved with diffusion imaging in the human body?
Oh, the biggest surprise, Dick, what do you think?
Well, I would say it's durability. So if you look at the early days of MRI, I mean, the exciting thing there was that even lowly MDs like me could be involved in explaining why certain things looked the way they did, because the people who introduced the modality had no idea what a lot of tissues would look like, what a lot of disease processes would look like. And so we had this opportunity to do simple kitchen table physics and learn about almost, all kinds of things and explain what we were seeing in the images. And at that time, if you look back to see what we were enthusiastic about, many of those things that we kind of explained and then were enthusiastic about didn't really turn out to be all that important in the long term. The interesting thing about diffusion is like almost from the beginning, it was there, and then in imaging, and then it's continued to build. So it's the durability of it as a phenomenon and what it can tell us.
Yeah, I think it was the surprise of getting the first set of cell-shielded gradients for our little animal system. That surprised me. I spent days trying to measure eddy currents with self-shielded gradients that Pete Romer had built for that little scanner of ours, and couldn't find eddy currents, just couldn't. And it was amazing. It was like having a Tesla for the first time. It's like, this is amazing. And being able to ramp up to being able to ramp up to B values to 1,500, 2,000, 3,000, even in those days, was remarkable. And then finding the weird stuff about, ischemic tissue. And of course, it seemed like anything you bought at the grocery store had anisotropic diffusion. And that was a surprise. And now we see, of course, that 90% of our scan time at Lucas now is funded by people doing fMRI and DTI and the correlation thereof. And it's just remarkable. I can't imagine how this is going to become. I'd like to see the clinical implementation of DTI on an everyday basis like we see for DWI. I want to see the DTI become that useful tool that's going to become part of every neurological protocol.
So perhaps I could ask everybody to chip in here, because I think, Mike, your observation of the reduction of diffusivity and ischemia has become, globally important for medicine, early intervention in stroke. It's probably one of the most important applications of diffusion MR. But the rest of the stuff hasn't yet made it. Denis, it looks like, Denis is getting ready to.
I hope it works. Yeah, I had prepared something also about what I was doing. Do you hear me? Do you see the screen?
Yes, we can, yeah.
Good. Yeah, so that's me when I had more hair back in 1984. As I said, I was a student, and I was working on the first French MRI scanner, and also on a 2 Tesla spectrometer. And because I was also a radiologist, my question is, we got beautiful images, but we had no idea about the nature of the tissue underneath. We could not access a microscopic scale. And this is how I came up spontaneously with the idea that if we can measure diffusion of water molecules, they will be like spies, giving us some information about the tissue. And of course, there is huge literature on Brownian motion and diffusion. The main one is from Einstein, which I read his papers many times. And I was so happy to see that according to the Einstein's relation, we are in exactly the right scale at the microscopic scale. On the other hand, looking at the literature, there were many papers which had been published about the effect of diffusion on the NMR signal without localization. But we have to recognize that diffusion was an artifact, a nuisance at the time, because the field was not very homogeneous. So as Mike and Dick said, the T2 was shortened by diffusion. So it was an artifact. I think we have to recognize that Stejskal and Tanner inversed the algorithm by showing that indeed diffusion was a useful parameter, but they could only study dead tissues. So this, in fact, zoom on my screen. You see, I was playing with hand echoes and diffusion and so on. So this is how I mixed up everything. So from Einstein to imaging principle and NMR, diffusion NMR, to these French papers that you mentioned. By the way, Paul Letterbourg and Peter Mansfield both mentioned that with imaging, we could have access to diffusion, both of them, but they never did it. They just said it was possible. So this is how it worked. So I implemented the sequence on the French scanner. The problem was that we could not apply the Stejskal and Tanner equation. I think this is a very important point because it took me years to convince some vendors that their calculation was wrong. This is how I introduced this very simple equation where B, what we call the B factor, in fact, was for my name. So not many people know about that because it was a little bit complicated to show everything, the double integrals with the cross terms and everything. And the second big the idea that I introduced there was that we don't get the diffusion coefficient. We get what I call the ADC, apparent diffusion coefficient. First, because diffusion was not free. We use Einstein's and Stejskal-Tanner equation for free diffusion, but what we see is not free diffusion. But also, I had the intuition that we also were sensitive to blood microcirculation, something I developed as IVIM and the pseudo diffusion coefficient. So this is, everything came at the same time in 1984 to 87. This is the very first abstract that published in London. The image is my brain. At the time, it was possible to do that. Today, I think it's not possible anyway. And I knew, of course, the work that Mike talked about, but my concern was that only a relative diffusion coefficient was obtained, as you showed at the end of the paper, D over W. And also, if I'm not wrong, they use a slice selection gradient for diffusion encoding. And if you change the slice thickness, of course, that's an issue. But what was very interesting is that at the time, we used slides, not PowerPoint, of course. And I was sitting in a room, putting all my slides in order in the carousel. And next to me was sitting another guy, Dieter Merbold, who was doing the same thing on an abstract on diffusion also. But he used the steam sequence. It was not quantitative at the time, and he had hand diffusion images. And at the same meeting, Taylor and Bushell also, I know that David is not here today, but I'm sure he will be there later. And they used, as you showed, a steady gradient pulse. It was not really pulsed, I think. And they showed the hands egg phantom. So that worked very well, but that was not enough for me. My goal, of course, was to go clinical as fast as possible. So immediately, because I was also linked to a hospital, in a pediatric hospital with CAM patients. And the images, we were so amazed by what we saw, something we could not see with T1 and T2. So this was shown at the RSNA. I was, again, a student, so to get a magna cum laude on the first presentation at RSNA was a big thrill for me. And this was the very first paper published on diffusion MRI in a clinical setting. And again, with the idea that perfusion was probably playing a role. This was showed a little bit later, well, 2 years later, with another abstract for the ISMRM, where I showed that we could separate the true diffusion measurements from the perfusion measurements. And this is the time when I moved to NIH, and Bob, I'm sure he will talk more about it. I made, the way to go to clinical work was, at the time, the B values were like 100, 200 on lucky days, and it would take 5-10 minutes to get one B value. So thanks to Bob and the gradient coil he had made in the EPI sequence, we could get images very quickly at high B values without motion artifacts. But I think the guy who made it through the clinical world is Mike. I mean, without his paper on acute stroke and the drop of the ADC, clinicians and manufacturers would not have been so much interested in implementing diffusion MRI. So I think Mike is a person who really made the difference in order to switch from basic research to clinical applications. And the second discovery, I think it's a finding, it's a discovery that he's made, because that was not done before, that in the brain white matter, diffusion is anisotropic. I think even Stejskal and Taylor had not realized it. And to show all of that, with Bob Turner, we organized the very first ISMRM workshop on diffusion MRI. I invited even Stejskal, and he was such a nice man. He passed away some years ago, unfortunately. But it was so nice to talk to him, and he was so proud to see that his work had some clinical significance. So now going to this anisotropy that you mentioned, and in fact, Mike has discovered, very quickly, I consider that we should reverse it. Instead of measuring diffusion in different directions and looking at different ADC values, we should do the opposite. We measure diffusion in different directions, and we guess what is the orientation of the fibers. So this was, in fact, the very first paper we published on fiber orientation color mapping. There were two directions, for instance, vertical and horizontal. So I showed this at NIH meeting in 1990, and this is where our pathways with okay, it's working. With Peter Basser, this is where we met. Peter Basser came to me. He was very, I mean, he could correct me if I'm wrong, but he said, do you know that diffusion is a tensor? And of course, I said, I know, but I don't know how to measure it with MRI. And this is how our collaboration started, extremely successful, because we've got these two major papers, which have been highly cited. And you see that over the between 1985 and 1992, where we developed this, we had many patents. In fact, I think about 5 patents about diffusion and IVIM. But the DTI patent changed everything because it was like a generalization of the diffusion concept. So this was also published at the ISMRM. As posters. I think Peter was not so happy with that. He might talk about it, but it was the very first time we introduced the B matrix. So it was a promotion for the B factor. It became a B matrix. And also the idea is that we could use DTI to do tractography. And next week, Peter and I will receive a quite prestigious award in Germany for DTI. So just to finish up my slide, so I don't, and that it took 30 years. So the conclusion is that many years have passed. Diffusion MRI has been extremely successful, from acute stroke to cancer, brain functional MRI, and recently I introduced virtual MRI elastography. But I guess there are hundreds of people who have contributed to the field. But my advice to young students, that if you are convinced you're right, keep going and don't give up. You know, I've been discouraged so many times by prominent people who told me, no way, you cannot measure diffusion because of immersion. No, you cannot do it. And those people, some of them, many of them, in fact, I don't want to give the names, switched to diffusion MRI some years later. So you see, my message here is that if you think you're right, go ahead. So that's all. Thank you.
Parfait. Merci bien. If I could ask you to stop sharing your screen, please, Denis.
Yes, yes.
Thank you so much, Denis. And I think there was just didn't quite get a chance just to say thanks to Mike and Dick for their contribution before Denis' slides started. But particularly just wanted to observe that Mike has got up at the crack of dawn at 7 a.m. in California, and Denis stayed up until the end of the day, until 11:20 to midnight now it is in Kyoto. So thank you so much for that. And you know, it's been a long career for everybody. Denis, you mentioned the patents. For you, what has been the piece of work or the development that you've been most proud of along that pathway? Is it that very early demonstration of diffusion, or is it a later application?
Well, I've been proud of everything, but I think the very first images, I mean, I didn't say, but I started in the liver first. And it was a disaster because we had no motion correction. So I quickly changed to the brain. But to see those images in those diseased, they were children, you know, and with tumors and so on, to see this contrast that nobody had seen before was really something I remember. Yeah. So I think it was the fact that I implemented a new technique, but within a few weeks, I got results that were largely beyond my expectations.
Yeah, brilliant. And you made a comment, just a word of further word of encouragement to trainees, that Peter was disappointed with his poster, his work only being presented as a poster. I think that really sends a very clear message that some of the best work at the annual meeting is actually contained in the posters. And
they may even be rejected. They may be even rejected.
They may be rejected, yeah. And I always see it as the best place to have a deep conversation with people about your work, more than the 3 minutes of question time. So,
Sure.
you know, poster bill does not mean anything. And you know, look at the history of our field, and particularly diffusion tensor imaging. Started out as a poster and is still going strong. So Denis also did a great job of introducing our next two speakers. He mentioned how Bob Turner's input was critical there. And so with one eye on the time, I'm going to ask Bob to tell us a little bit about his time after moving from Nottingham, which was, just as a slight anecdote, was where I did my undergraduate degree. And my final year project was to look at a paper by Bob Turner on the target field approach. And so that got me into MRI. So thanks to Bob, that got me into this mess as well. So with that, over to Bob.
Well, yes, well, thank you. I'll share my slides now, but I'm not going to show all of them. Where's it gone? No. Have we got it? How to do that?
Okay, so I've got Bob's slides, and just give me a shout when to advance, Bob.
Okay, well, what I'm going to be describing is what I consider I contributed to this development of diffusion imaging, which really came about because of wanting to do something a bit different. I actually wanted to measure perfusion, and after I'd come to Nottingham and learned how to do MRI and built my own scanner, as I mentioned before, I felt that maybe the way to measure perfusion was to use diffusion gradients to sensitize the image to the movement of blood. So I came up with this kind of in parallel with Denis and Bihan, and but I actually got it wrong slightly. But I still needed to get the technique of diffusion-weighted imaging going. And can I have next slide, please? There we go. But it very quickly was clear to me that if you actually wanted to measure diffusion in a living human brain, you had a problem. And the problem was that the gradient pulses not only sensitize the image to the movement of blood and the movement of water molecules in the tissue, but they sensitize the signal to the bulk motion of the object. And if you're using a multipulse sequence, then you have perhaps random different movements between each pulse, each acquisition, which means that you end up with terrible motion artifact if the head is not kept extremely still. It's extremely hard to keep the head that still. So I had to think of a way of getting better images without motion artifact. Next slide, please. And in 1986, long before I actually was able to implement a diffusion-weighted echoplanar sequence, I proposed a solution, which is to form the MR image after 1 RF excitation. And of course, the way to do that was EPI, and the way to make EPI sensitive to diffusion was to put in diffusion gradients, either around a 180 pulse or using bipolar gradients, which is another nice way of doing it. And I proposed to do this at a NATO Advanced Study Institute on Cerebral Blood Flow, which was held in L'Aquila in Italy in 1986 in June. And I was actually there as a student on this particular study institute rather than a lecturer. But it so happened, and this is a fascinating twist of history, that this was the year when there was the bombing by terrorists at the Fiumicino airport in Rome, which is where the most of the people who had wanted to attend this meeting had to fly through. And quite a number of the staff in this Advanced Study Institute were coming from America, and most of them decided to cry off. Their wives in particular said, "Don't go to Italy, it's too dangerous." But the one person who did come from NIH was Lou Sokolov, who was one of the most brilliant brain scientists I ever had the pleasure of meeting. And he was at NIH at the time, and he did give his lectures. And so that was one good thing. The other good thing was that I got promoted from being a student to being a lecturer in this course. And I had to improvise some transparencies very quickly to describe what I wanted to show them, which is what I wanted to show them was diffusion-weighted EPI. So that was an interesting turn of events. And so I gave my talk, and next slide, please. And of course, 2 years later, I finally got the chance to implement diffusion-weighted EPI. To my great pleasure and delight, I found when I moved to NIH in May 1988 that Denis Bihan was already there. I'd been following his work for some time on diffusion, and I knew he was the person in the world that I most wanted to talk to because of IVIM and also getting DWI right without motion artifact. So at NIH, I was actually there, I was employed there to develop gradient coil design, which is, as Derek has just mentioned, what I've already best known for is techniques, the target field method for designing gradient coils and shielding gradient coils. But what I really wanted to do was study brain function. And so I built, or I had my head gradient coil built for the 1.5 T Cigna. And I was implementing echoplanar imaging all over the place. So when I first arrived at NIH, the scanner was going brrr, brrr, brrr. When I left, they were all going beep, 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 beep.
You can hear the remake of Bob's EPI sounds on the next available album.
Next slide, please. So there's the home-built Z gradient coil. And this produced a very high gradient because it's very small, just fits around the head, just one axis. But it gave us really nice echoplanar images. And next slide, please. And this was the publication of the first diffusion-weighted EPI sequence, which was, as I say, presented in 1986, actually published, the book was published in 1988. But there you are, you've got the diffusion-weighted sequence, the lower figure there. And that's what really got, as Denise says, it made an honest imaging sequence out of DWI. Next slide, please. There it is again in the radiology paper 2 years later. Next slide, please. And those are the kind of quality images. They were really reliably motion artifact free. And next slide, please. And from these, you could actually make quantitative measurements of diffusion, the apparent diffusion coefficient in the brain, and you got lovely consistent results across subjects, as shown in that graph there. So from there on, of course, we went to wind the clock years and years later, and when I moved eventually to the Max Planck Institute in Leipzig, we were able to do very, very high resolution DWI, DTI, at 7 Tesla with 800 microns resolution, and produce images like this. And of course, that's the amygdala we're showing there, showing a few scattered fiber tracts. It's mostly gray matter, but you can actually see the complex pattern in a structure like the amygdala. So yeah, that's basically what I would like to say in this wonderful gathering. Tremendous inspiration from Denis Bihan for many, many years, and fantastic conversations with Peter Basser. And also, I have to say, what a tremendous pleasure it's been to work with Mike Moseley over these years, and particularly in those early years when he was doing those fundamental work on stroke in cat brain and the anisotropic diffusion in cat brain as well. It was just a great time to be alive then in the late 80s and early 90s, I have to say, and of course thanks to my collaborators. So thanks for this.
Thanks, Bob. And yeah, it's really great to see the actual original hardware. And I'm going to challenge the younger members of the ISMRM to sample your imitations of EPI and digitize it and use it to form an MRF sequence, and let's see the kind of images we get out of it. I think that would be fantastic. We could call it the Turner sequence. Well, time is running away, and I'd like to get everybody's reflections at the end. But Bob, what you've really painted a picture here is a real family of diffusion images, and you mentioned the final member of the family to join today, which is Peter Basser. So again, last but not least, Peter, congratulations to both yourself and Denis on your award you'll be getting in Germany. That's fantastic news for you and for the community. And so you're going to tell us a little bit about DTI and tractography.
Thank you so much, Derek. And I feel the same way that Bob did about, having wonderful collaborators. Having Bob down the hall in the biomedical engineering program and Denis in the clinical center was just such a gift. In retrospect, I just can't believe the wonderful coincidence that we were all there together. I just wanted to provide a little insights into DTI and tractography. As Denis alluded to, the concept of diffusion imaging was clearly articulated by Paul Lauterbur, where he basically said that MRI could be combined with NMR spectroscopic techniques, and he specifically included diffusion coefficients. So this is, I think, a very important contribution we sometimes forget. Standing in front of Denis' poster with Philip Dueck and Bob Turner, I started to think about measuring anisotropic diffusion using a tensor, which wasn't the approach that was being used. And I went back to my lab and started to concoct this memorandum that I ended up sharing with Denis and Bob and Philip and Brad Roth eventually in November of 1991. And basically it was a playbook for how to do DTI. It described the diffusion tensor, the eigenvalues and eigenvectors. By the way, this is all found in Derek Jones's wonderful DTI book. That this is not a product placement, but I should mention that this is in one of the later chapters.
My family would be grateful for the royalties, Peter. Thank you.
I don't expect any kickbacks there.
Okay.
And it describes the principal axes and the diffusion coefficients, the eigenvalues, and it actually shows what I think is the first representation of tractography. I'll discuss this in a moment. The mathematics to produce the diffusion ellipsoid, which represents features of displacement distribution. And some of the scalar invariants that you generate from the tensor, these are quantities that are intrinsic to the tissue, like the trace, which has become such an important parameter in radiological applications, and even anisotropy measures that could be represented by ratios of eigenvalues, which replaced ratios of DWIs or ADCs, which were not intrinsic. And I guess, Denis explained to me in the meeting that nobody really knew how to measure the diffusion tensor. And I think that I had the presence of mind to ask him and Bob how I would go about doing that if I wanted to learn more about NMR. And they both said, read Stechkoll and Tanner. Bob said, everything can be found in Stechkoll and Tanner. And to some extent, he was right. And I buried myself in the stacks in the NIH library, and I eventually came across some articles by Stechkoll and came up with a relationship between the signal attenuation, the diffusion tensor, and something that I called the B matrix, generalizing the B factor that Denis had talked about. And now jumping ahead, these are the first tensor measurements I'm aware of diffusion, the diffusion tensor in water. And it shows a nearly isotropic tensor that has diagonal elements that are more or less the same and off diagonal elements that are much closer to zero, but not quite. And it was bothered me quite a bit that we didn't have a real isotropic tensor for water. And I wondered, if we can't do it for water, how are we going to be able to look at other tissue like skeletal muscle or brain tissue? Eventually, it came to SMRM, or ISMRM now. And Denis had mentioned that I wasn't so happy about where these abstracts ended up. They were both relegated to what I call Posterville. But the one on the left was basically the mathematics and the experimental design for measuring the diffusion tensor in the laboratory coordinate system. And then the examples were diffusion in water and diffusion in skeletal muscle. And on the right was basically the memorandum that you saw with the additional data of skeletal muscle, where we rotated, Jim Mattiello had done these studies, actually the experimental data collection, where he rotated the skeletal muscle about 43 degrees along the z-axis and measured the diffusion tensor twice. And you can see how the ellipsoid gets rotated with the principal axes of the tissue. So this showed that the tensor elements changed significantly when in one coordinate frame to the other, but the invariance and the eigenvalues don't. And this also, discussed things like the scalar invariance and other things that I've described already. So let me, in the interest of time, let me skip through. I did want to mention a little bit about tractography. In that same abstract from 1992, I wrote that one could envision 3D fiber maps or diffusion ellipsoids displayed in each voxel, connected like link sausages that follow fiber tracts. So this was really the first publication I'm aware of where anyone was talking about tractography. I didn't show an algorithm for doing it, but it does. This is basically constructed. This was from that same memorandum from a fiber. Sorry about that. From a velocity field that's well known in fluid mechanics, which was what I got my training in. So, but it took till 1997 to formulate and put together a proposal, an algorithm, a methodology for doing fiber tractography with DTI data. And it's basically a simple algorithm that follows the direction of maximum diffusivity and models a fiber tract or nerve or axon bundle as a space curve, and the slope of that space curve at any point is given by the eigenvector associated with the largest eigenvalue. That's the direction of maximum diffusivity. And you solve that system of differential equations and you can elaborate a tract. And DTI, as we know it now, isn't that dissimilar from what it was, what I proposed back in the day, and eigenvalues, eigenvectors, Carlo Pierpalli's deck maps, diffusion ellipsoid images, tracks. So I think that covers the gamut, but I think that's kind of where I thought would be helpful for people to find out a little bit more about DTI and tractography.
Brilliant. Thanks very much, Peter. And if I could ask you to stop sharing your screen, that would be great.
Okay.
So yeah, thanks so much, Peter. And there was a comment made by I think it was either Dick or Mike earlier on, and I was just starting to ask. I was commenting that Mike's drop in the ADC was probably the most clinically used aspect of diffusion. Since that time, of course, there's been many developments, and Peter, you've just shown us, the tractography, and we see beautiful delineations. It's now used for pre-surgical planning. And I guess I just computed there's been another 28 years since 1994. We've seen even more developments, but it hasn't really, there's not been as many sort of uptakes into the field. I think maybe it was Bob who said he wanted to see DTI, or maybe it was Mike, adopted into the clinic. What do you, and I'll ask everybody this, not going to put anybody on the spot, but please chip in. What do you see as the biggest hurdle to adoption of, some of the more advanced measurements that we're seeing in the clinic? And do you think there's promise? And, what do you think the technological hurdles are that we need to overcome?
If I may say something, DTI is especially useful in the brain, but diffusion MRI now is used mainly outside the brain. Body diffusion MRI is a very important field. And in the body, the anisotropy is not so important. There are a few papers on DTI in the breast, in the kidneys, in the muscles, where they are somewhat anecdotal. It's not really used clinically. So I think that's probably why we have this bias. If we consider body diffusion MRI, DTI is not such an important technical aspect. But in the brain, I don't see how we could publish anything in the brain without using DTI. If people measure the ADC, if they don't say how, in which direction they measure it, it's worthless. So in the brain, I think even if people don't mention it, I think everybody is using DTI. I mean, if they use a trace ADC, they use DTI. That's what I feel. So that's my small comment on it.
Well, Derek, I think maybe Dick Ehman may have some perspectives on this. There may be some issues having to do with reimbursements that may be driving some of the uses of these technologies, at least in the United States, in clinical applications, because the CMS does a kind of cost-benefit analysis, and they come up with recommendations about which techniques should be reimbursable and which aren't. And I think sometimes these techniques aren't reimbursed in many procedures. Dick, can you add that? Because you're a card-carrying radiologist and you know about this stuff. I'm just a pretender.
Oh, thanks, Peter. Well, first of all, I really enjoyed the amazing stories that we've heard in this session here, and I'm just hats off to all of you in this area. I think that Mike brought up the issue of transition to clinical practice, and Peter, you're right. There's to some extent, I think that there may be a reimbursement issue, but generally speaking, in MRI in the US and CMS or in the CPT system, there really isn't a coding for particular procedures. So as long as if we found, to the extent that diffusion, this phenomenon in all of its forms and all the ways it can be used, can be shown to be useful in clinical practice, it will be used, and I think it will be reimbursed as part of the anatomic exam that's billed for. So I think it gets back to a little bit of what Denise said, which is, if you think about it, the vast majority of the use of this phenomenon in diagnosis is the simplest diffusion-weighted image. That's used every day by almost in many exams. Maybe that's telling us something about what ends up being useful sometimes in clinical practice. And obviously, it's had the whole phenomenon has had massive advances in research as a result, understanding of disease. And I'm not a neuroscientist, so the tractography and so on, I think is just amazing. But we aren't using, for instance, measurements of ADC in very many applications, and that probably relates to the difficulty of using quantitative techniques. And we talked a lot about that. And there's a social in radiology, there's a reluctance to use quantitative measures, but also there is the problem of cross-platform compatibility. I just want to point to one thing, though. I think that as we talk about non-brain applications, the whole area of cell cytometry, I think, is an area that probably has a lot of applications, with diffusion measured by diffusion, probably has a lot of potential. I know there are many people working in that area, but probably an area, in my opinion, that probably has some real potential for quantitative and clinical practice.
Yeah, I agree. That's completely right.
We're talking about the past, and we're talking about how all this started. And I was illuminated on Friday by the young generation of MR physicists, the young generation that is going to take this even further. And what I've begun to see is that, from now on, ADC, DWI, DTI, it's going to become issues of fingerprinting. MR fingerprinting for diffusion-weighted behaviors is so powerful, coupled with AI, coupled with case-based sampling and Q-space sampling. I'm just amazed. And for those watching this, if you get a chance to talk or to look at some of the work by Cowan Setzenpop, he just came to Stanford from MGH, and he just blew us all away with his new vision of diffusion, because quite frankly, DTI or any MR sequence, and Dick will tell you, radiologists are not going to want to do that if it's not less than 4 or 5 minutes. And so you put together the fingerprinting and all of the tricks that we have today, it's a whole new world. And I'm just so excited by what we're going to see in the next 20 years.
I mean, Mike, I absolutely agree. I have the pleasure of also working with Dan Marr and Mark Griswold, who are also doing diffusion and fingerprinting together. And the acceleration factors, it kind of, you say, yeah, but your acquisition took 3 minutes, but our 45-minute acquisitions were still good fun. So you just mentioned the next generation, and I'm in no doubt whatsoever that people will be watching this recording years from now and still feel totally inspired, because what the present company has done here has basically circumvented massive technological challenges. So first of all, how can we quantify T2? Then we find there's the diffusion effect. So we actually go ahead and we work out how to image that. Once we've made the first images, how do we make it robust with EPI? Then we observe that there's anisotropy. How do we characterize this? Well, we need to estimate a tensor. How do we do that? Then we do it. How can we reconstruct fiber pathways? And it's all been done. But in each of those times, before that problem was solved, it was a frontier where we would look and say, but how do we estimate the tensor? Or how do we make these measurements less sensitive to robust motion? So again, I'm putting you on the spot. What do you think are the current frontiers that you think, we don't know how to do this yet, and if some young, energetic soul can leap forth and come up with a solution, that's going to be the game changer? Maybe it's the MR fingerprinting, Mike, and getting that down and getting the acquisition time down. But I wondered if anyone has any additional things they'd like to put onto the list.
Well, I think what Dick says is quite right. Radiologists don't like too much quantitative imaging. They want to see. They don't want to use numbers. So we have a revolution to prepare in order to convince them that they have to switch to quantitative imaging. And then for diffusion MRI, the big problem is that it's standardization. Everybody has his own protocol. The B values are different. And because of non-Gaussian diffusion, that means the ADC values are different. So it looks like, for somebody who is new to the field, that the values are not reliable because you find different ADC values across centers. So we need quantification and standardization so that people can trust what they measure.
Mm-hmm. Yeah, quantification, it's the revolution.
Yeah, I'm French, don't forget that.
Vive la revolution. Bob, I think you were just about to raise your hand.
Yeah, I wanted to comment on the usefulness of tractography in neuroscience. Really, this is the issue. It got very popular for a while, and then it got less popular in meetings like human brain mapping and so on, simply because of, well, partly because of what Derek and I published in our do's and don'ts of diffusion imaging paper, which has been incredibly widely cited. I think people took a lesson from that, that there's some things that you can't deduce from what we have been doing in terms of DTI and so on. We need to have imaging which tells us a bit more. And of course, the work of Van Wedeen and others has shown us that we can actually look at crossing fibers and so on. I think that area of research is still not yet mature, and what it really needs is not so much an input from MRI, but an input from neuroanatomy. And that input from neuroanatomy is coming with much, much better ways of looking at how the fibers organize themselves in the brain. We're learning more and more, even now, about U fibers and the way that fibers cross and so forth. I think that once we have this anatomical knowledge, which it's an incredibly complicated picture. I mean, when you do fiber staining tracking and so on using conventional methods on animal brain, you realize just how complicated the wiring diagram of any brain actually is. Once we get a little further along there, then we can much, much better interpret the MRI data. And I think that ultimately will lead to better models of how the brain works, but also better algorithms for how radiologists, clinicians can actually interpret the data. So I see further progress as feasible there. Of course, putting this together with machine learning and such techniques for handling vast amounts of data, I think that there is a road forward which will tell us a great deal more about brain connectivity.
Yeah, thanks, Bob. And Peter, you've been, really pushing things like cell size measurements and looking at exchange and, multiple dimension, combining diffusion with relaxation. Again, what do you see as the biggest obstacle that we need to overcome? And, do you think it is that we need to start combining diffusion with other dimensionalities, for example?
You want to know my dream? Oh, sorry. Yeah, go ahead.
Peter, yeah.
Oh, well, that's certainly been, a focus of our group for quite a while now to, not have blinders on and think that our own, diffusion measurements would provide all the information we need. And I think there has been a lot of progress made in, looking at things like astrogliosis and diffuse axonal injury, for instance, using T1, T2, and diffusion information together when they're correlated. That's certainly, I think, a promising direction because you're using complementary methods that provide complementary information. We certainly looking at exchange and restriction together is important, and being able to separate them. I think looking at many different length scales is very helpful. We're working with the Mass General group on the Connectome 2 project, and being able to look from the Jeff Lichtman scale of SEM all the way to the whole brain is very useful, because there are tremendous constraints, and there's a lot of information provided at different length scales that force you into assuming or challenging your assumptions sometimes that allow you to make progress. I think resolution is becoming an issue in our hands. We'd love to see higher resolution images, and there's a tremendous amount of partial voluming that still goes on in the cortex and in white matter, with a 1.5 millimeter voxel size. So being able to shrink that down and have more pure voxels, I think will make a lot of things snap together. And that's really an engineering issue, which I think we can solve as a field. I agree with the rest of you who've talked about the power of AI and other computational methods for not only improving experimental design, but the interpretation of acquired MR data and speeding up and improving the quality of the data that comes out. I think these are all very important. That's pretty much a summary.
Great, thanks, Peter. And Denis, I think you were just going to chip in.
Yeah, just my dream is it's good to have the connections, but what I want to see is the flow of action potentials along the fibers. So that may not be possible with MRI. I'm not too sure. I'm thinking about it. But combining DTI at high resolution with other methods might help. So we want to see the flow of information through the brain connectome, just not the static brain connectome.
So I think you heard it.
I don't know.
Here, you heard it here. The Libyan Prize for Action Potential Imaging.
Yeah.
Well, one of Paul Adderbur's students, Vika Scalani, was trying to do that many years ago, stimulating sciatic nerve, I believe, and then trying to see changes in signal associated with propagation. And I think it's a dream for a lot of people, and I agree with you. It would be wonderful to be able to see brains, function at the time, in the time of thought, at the time scale of thought, and we're not there yet by any means.
Okay, so I am very aware that it is way past the bedtime of Professor Libyan, and it's already getting into morning for our colleagues on the west coast of America. And I also want to keep our powder dry, because there will be an opportunity for people at the ISMRM to come and actually meet the legends, either in person or virtually. And there'll be two opportunities. One is on Monday, the 9th of May at 5 p.m. That will be on Gathertown. And there'll also be Wednesday, the 11th of May at 2:30. So what I'd like to do, rather than me have the last word, I'd just like to ask each of our speakers if they would just like to give us reflections, words of advice, observations. So perhaps I'll start with Mike.
Yeah, if I could see anything in the next 30 years, well, I will not see that. But I want to focus on a couple things that would truly excite me. Number one, aquaporins. Is the ADC actually a measure of the aquaporin membrane channels? And that particularly hurts me that, this is the whole reason behind the ischemia. And I think it's aquaporins. The second thing is we've always talked about the ADC or a tensor map being a static image. It's not. Diffusion is fluid. It's dynamic. And we don't do dynamic diffusion. Why is that? And then finally, of course, someone will point to a Rosetta Stone that will link diffusion MR with cognition. Still waiting for that. Thank you.
Awesome. Thank you. Dick.
Well, just maybe two quick points. One being, I think that on the scientific side, I think the whole area of separating out the extracellular matrix from the intracellular phenomena and the extent to which diffusion can help us with that, I think is important and needs to be, we need further work in that area. Then finally, it should be obvious to the younger members of our community as you listen to these giants here that they all have been passionate about what they did, and have had a lot of fun in the process. And I think that those are important parts of a successful career.
Indeed. Thanks, Dick. I think Denis.
Yes, well, just to go about what Mike said, linking diffusion MRI with cognition and dynamic ADCs, this is diffusion fMRI. So we've done it now for more than 20 years to look at changes in the diffusion when people are performing cognitive tasks, and it's working very well. Now we are working on a very sophisticated model at whole brain level, and we are able to wake up animals who are anesthetized by modulating the diffusion of water in some very specific parts of the brain. So we are now considering to do that in patients in a vegetative state, and diffusion MRI will be used to monitor the effect in a very specific basal ganglia. So yes, we are going on this direction.
That sounds fascinating. I look forward to seeing more of that, Denis. Thank you. Over to Bob.
Well, I definitely agree with Mike in the directions that he hopes that we can look. But in particular, I think the question of the dynamic quality of the fiber structure of the brain, the fact that there is a bootstrapping process of myelination going on in the brain, which is clearly obviously affects T1, but it also will affect the diffusion parameters as well. And so longitudinal studies in development, I feel, will be very important information in actually modeling how different brain systems work in cognition, because different networks in the brain will come on stream at different times as we learn things. And using the combination of quantitative T1 and quantitative diffusion, I think we'll be in a much better position to understand exactly what parts of the brain are working together to make things happen and how they're doing it. So yeah, that's where I would like to see progress.
Fantastic. Thanks, Bob. And again, Peter, please.
Thanks, Derek. Well, I think I'll take a change of course or tack in another direction. I think persistence is really critical in having a research career. Denis had, alluded to it, and not to get discouraged. And I think another critical thing to leave with, people who are looking at this, who are maybe a little younger, starting their career, is, the importance of mentoring. I was very fortunate that I knew Bob when I was starting to, do these early diffusion tensor posters. And in Berlin, I was quite discouraged when virtually no one came to those posters. And Bob asked me, Peter, how did it go? And I said, not well, Bob. He said, well, how many people came to your poster? I said, well, maybe 4 or 5 people came, and there was very little interest. And he said, oh, that's great. He said, this is just the first year. He put his arm around me and he said, this is wonderful. Don't worry about it. And, that really had a huge impact on me. And I want to thank him publicly for that, because, and I think that there are critical times in scientists' lives where you can really intervene and help some young scientists in particular, and help their careers and their buoy up their spirits, because they're really, it's a long haul, and I think you have to be prepared for it. And persistence does pay, no question about it.
this has just become a wonderful loving, and it gives me a warm, fluffy feeling inside at the end of the session. But I think Peter's comment there about mentoring, I just would really encourage the younger members watching this not to be intimidated to go up and talk to the people on this screen. I remember feeling incredibly intimidated by everybody's name on this screen. And the first time I met, I felt nervous, felt anxious. When my supervisor picked Bob Turner to be my PhD examiner, I thought it was the cruelest card that could ever be played, because I was so nervous, as Bob will attest to. But in the end, he was very kind. I ended up working with Peter, and Peter, again, I'd like to join the love in and say that, yeah, your mentorship to me was influential and helped me huge. So yeah, these scientists on the screen, they're eminent, and you may feel intimidated, but I know each of them has a wonderful personality and would be very happy to spend time talking to you. So please go and grab them and chase them around the conference center at the meeting. So as I say, you have a chance to come in person on the Wednesday at the meeting and meet some of the people here today. There's a chance to join the Gather Town to ask questions. And indeed, you can always, I'm sure, just send an email to each of these colleagues and with questions, and I'm sure they'd be happy to respond. So very conscious of the time, I would just like to say how much this has been a treat for me. I've thoroughly enjoyed it. I felt slightly anxious still. I wanted to do this service because it's a historic gathering of people that have been the history makers in our field. So I'd like to thank each of our speakers in turn. So Mike and Dick, Denis, Bob and Peter, thanks so much for your time. And yeah, look forward to seeing you in London, either in person or online. So thanks very much.
Yeah. Thank you,
Bye.
Derek. You've done a great job.
Wonderful.
Yeah,
Thank you very much, Derek.